Go away, dude. Go away. Morning. I feel like I bright lights on, and then starts watching something with my dad. With my dad. Hello. They're like taller than what? I separated these. Good job, Lauren. I wish I could find them. Traffic lights, probably so. Yeah. No.
Letters are out for the nursing.
Oh, yeah?
So we saw it in our mailboxes coming, so it's gonna be the longest day of our life. Yeah. They're like, they're like coming from the nursing line.
Yeah. Chapter 9 and 25.
Has anyone knocked on the sign sheet? While we're waiting to start on this, we get a couple more minutes, because there's stragglers coming in, or stragglers lining up or whatever. Does anybody have any questions about any of, chapter 6 or chapter 8 or anything coming up? Anything like that? Okay, while I'm here, figure it out.
Is there, you might have said, I don't remember, is there like this study for the lab?
Yes, so the Kahoots that we use, like, to review, in lab, I plan on posting those when we get a little bit closer to the review date for that, because I don't want people like working ahead on them. I want people to like have prepared for it beforehand, not knowing what the questions are gonna be.
Yeah.
Whenever we do this, they're not worth points or anything, but I still don't want people just like, oh, yep, I saw this answer. That doesn't help. In case you guys, when you took the test, didn't figure that out. So yeah, applying it, yeah. So I will post it, probably that week. I think that's paced in the same week. I think it is, right? Yeah, so,
Okay.
yeah, we'll do it then.
Is that gonna be like how we took this test, like in the lab? It's all
No. We'll kind of go over when we get closer, but it is like, and like a practical. So we'll be at stations, and we'll be like going from station to station, and you won't have to make slides or anything. I know I was threatening to make everybody do the microscopes on this test. I literally don't have the energy to put that together for this exam, but I'm not going to rule it out for the final. So keep that in mind. I will say this, I'm not gonna I don't want to get too pissy about it, because I'm just getting sick of getting on people over it, but we had that talk in the lab, and I said, do these things and be careful, or we're gonna do this. And then literally like at the next class, when I got out one of the microscopes to demonstrate how you're supposed to do it, it was not put away properly. And there it was at somebody's seat that was sitting there, like, put it away wrong. So I'm not gonna lie, I was pretty mad about it. They could probably tell. I mean, they kind of joked. It wasn't like big mad. It was kind of like, kind of mad. But yeah, so I mean, it's a battle that I just feel like I'm not gonna be able to win at this point. If there are egregious deviances from how we're supposed to do it, like it was on the 4, it just was the stage was all the way up. I'm not gonna get too pissy about it. Please try to put it away. If like half the class continues like work to be putting up the stage on was on the 4X, but if it's just every now and then random people just didn't quite get all the steps in, that's fine. But if I start seeing people putting it away with the 100 with the stage up, like I had been seeing, I mean, it was like more than one person in every single section regularly, every single class. That's going to be a problem. So I'm just going to try to keep an eye out for it. We only have today, we have the Candace 4, which is microscope. Then on whatever Wednesday, I guess, the next lab is exercise 8, which is doing plates and stuff with antibiotics, sure, but we're also going to be doing pond water, so that's microscope too. And then after that, the next week we have the eukaryote lab, which is also microscope. These are not as big a deal microscope because they're going to be larger organisms we'll be looking at, so we don't have to use the 100 on those. But for this one, we will this time. And when we get back, I usually have us do, so when we get back after the midterm, lab midterm, I usually give you guys more, typically give you more of your stock samples with the blacktop lids because we kind of run through them. People have been really good about it this semester, so I really appreciate that. Hopefully we have more of those tubes made in time for me to culture more of them for you guys. We have enough, you know, for the rest of the semester. Anyways, when that happens, I usually like give them a new label and then have you guys redo a gram stain on them and refigure out which one was your gram positive and gram negative. And again, if I'll be there to tell you if you're right or wrong, but I just want you guys to get that gram stain is a big deal in case you guys have to start to notice that. So that we'll probably just redo that again. Same kind of deal as we did the first time. It shouldn't be too stressful, but that will be probably the last chance that you get to work with the microscopes actively. After that, we will not be touching them until you look at them again in the final, because they'll still be there on the final. But
Using the microscope
after the gram stain after the midterm won't be used. Okay, anyways, I don't know what led me into that. There you go. So I guess I was talking about the questions on midterm, right? But yeah, so it'll be stationed. You won't be setting up any slides or like, I'm not gonna watch you do the microscope. I just, like I said, don't have the energy for that right now. It's just too much going on. Dr. Tim's trying to lead a revolution basically. Be supportive of it. I agree with everything, all his choices, but he's just one of those people that like gets it in his mind and he won't stop. So it was like literally this whole weekend I've been emailing back and forth with him and other important people in the background. Basically, so guys, for those of you who don't know what Faculty Senate is or haven't heard what Faculty Senate is, Faculty Senate is representatives from each of the schools. So we're talking about STEM, which obviously is where I am part of, that's where science is, health sciences, which is where nursing would be. And then we have human services, and just like the police and that stuff. And then we have professional studies, which is like, I don't want to say the BS, like English and stuff like that, right? So it's usually not, most of them not program like degree related. But anyways, my whole point is we have representatives from that, and then we have like a president and a secretary and like a parliamentarian and then former presidents that make up our officers. Well, Tim and I are both representatives for STEM. He and I are the two representatives for STEM, the largest school. So we're representing a lot of people. And we think that Faculty Senate, which makes most of the decisions for the campus, to be clear, the big decisions. I don't mean the little things here or there, like making sure these people get this and whatever, but the big decisions, like making sure we have a committee on campus that has faculty representation for dealing with students who are struggling with mental health issues or substance abuse problems that need support. And they canceled that committee, the last Faculty Senate, before we started. And so we're fighting to get that committee back. I mean, we're winning the fight. There's no argument about it, but like, it's just like dealing with the government. And they have not been efficient lately. So when I say we're starting a revolution, which I kind of believe in. So hopefully this will be a good change for those people who are staying with the OSU OKP system in OSU OKP's nursing program going forward. Hopefully our changes will affect you positively. That's what Tim and I are doing. I mean, we're really having we have come to Jesus meetings and in class. Tim and I are having that with Faculty Senate. We're literally bringing them aside in an informal meeting to basically tell them, like, I thought that you signed up to make this school a better place and not be lazy and just vote yes and not read things, because that's what happened basically. So anyways, we're getting called out. So anyways, it's stressful. So I don't mean to be like short with you guys sometimes or whatever, but like I'm stressed out and like in the background, and I'm putting everything I can into this class as well. Don't get me wrong. So whatever. So let's do this first, and then we'll move on. It does this stuff. Just the reason I'm even bringing it up, I'm not trying to give you my problems, but it does affect you. So maybe not immediately right now, but it absolutely does in the future. So. All right, what type of biological macromolecule are restriction endonucleases? That's that word that I feel like it's wordier than it needs to be. Right, so yes, these are proteins. ACE at the end, anytime you see ACE. So we've heard about oats, like if we're talking about sucrose, fructose, lactose, OS means sugar, right? ACE means enzyme, and all enzymes are proteins, 100, okay? So that's how we know that just the ACE there. If you didn't know the rest of it, that part gives it away. But those are protein enzymes that are going to be cutting DNA at specific sequences. All right, there you go. I didn't get as specific as this is probably going to, but restriction endonucleases, what are they? Yeah, this is actually really general. Right, so these are molecular scissors. They are enzymes that are cutting literally the covalent bonds of DNA, like between the nucleotides, not across the strand, those hydrogen bonds, but we're talking about between in the actual strand. Cut the covalent bonds. All right, true or false? All restriction enzymes create sticky ends. Remember this. It's a pretty good depiction of what the sticky ends look like and how they work. Oh, it's a little animation too. I like that. Sometimes I actually put good pictures on here, guys. All righty. So they do not all create sticky ends. Now, the ones that we are most interested in typically for engineering are the ones that create the sticky ends. That's the ones we focused on, but they can create blunt ends as well. I'm probably not going to ask you too much about that on the exam. I mean, that's probably going to be referring to the sticky ends, just saying, but you know, cover all our bases. So CRISPR-Cas9 is a protective system in bacteria that can be used in genetic engineering to cut just about any sequence. Is that true or false? All righty, so this is true. We can manipulate it, and we are in the process of learning exactly how that can benefit humans. But we can manipulate to cut in just about any sequence of DNA, as long as we know the sequence. All right, PCR is a technique used for what? What is the purpose? What does it do? In case this is a picture of it, by the way, if this doesn't give it away, should kind of. All right, so this is the amplification of DNA in vitro. So if you guys get to a question like this, because we didn't deliberately go over in vitro, in vivo, in situ, anything like that. So if you get two words like this on the exam that we didn't explicitly put in the study sheet, you can always ask what this means, and I don't mind telling you. Okay, so in vitro means you have taken it out of, not in a body. It's like using a blood sample or something. You're outside of somebody's body dealing with it. In vivo means it is actively happening in a living creature. And then in situ is like taking a tissue sample or something that might still be technically living and is a representation of a living thing. And you could even look at those things in a living body. It's a little bit more complicated. But yeah, that's the idea of it. So it's kind of the in-between though. So yeah, in vitro and in vivo are probably the bigger ones I'd be familiar with as far as science. And eventually you guys will probably talk about some of that stuff later on in medicine. So which of the following is not one of the stages in a PCR cycle? There's only 3 steps. This is a term recognition question. Right, so this one is a little bit of the on the tricky side. So the one that is not part of the PCR cycle. In the PCR cycle, we have the heating of the DNA, that's called denaturation, separating the strands. Then we have the annealing of the primers. And annealing the primers is sticking them on to their complement with just the hydrogen bonds, okay? And based on their complementary sequences. And then we have extension or elongation by adding on the nucleotides. So what does ligation of the primers mean? When we talk about ligation, this is referring back to when we learned about DNA ligase. Ligation is talking about creating covalent bonds that are going to permanently attach things to something else. And we're not permanently covalently bonding these guys. In fact, we can't. That wouldn't make sense because later we're going to have to reseparate the new things and make copies of them. And then they'll be new, and then we'll have to separate those over and over and over again. We need it to be hydrogen bonds, not covalent bonds, really. So that's what this is more or less getting at. But don't forget about DNA ligase in DNA replication. Okay. What are plasmids made of? What kind of, macromolecule are they? I like this picture. It's an okay representation of kind of what's going on in there. Okay, so plasmids are made of DNA. The plasmids themselves are made of DNA. And they're little tiny circles of DNA. Comparatively speaking, we saw in the picture, there's a big old squiggly mess just hanging out in the bacteria, right? That was its chromosome. And I've told you guys, that's one big circular chromosome. It's just all kind of squiggled up in there. And then we saw these little tiny circles around the outside, and those were the plasmids. So plasmids are significantly smaller, little circular bits of DNA that have extra genes that can be beneficial, but they are not essential to the survival of the organism. So they are DNA. They do code for protein, but they are themselves DNA. Right? That's when a lot of people get tripped up on. If the plasmid contains a gene from another organism, it is called what? Right. So it is recombinant DNA. You can just think about recombinant, recombine. We've combined two things together that normally aren't together. And that really is all it's referring to. So plasmid has stuff from another organism. It'll be called recombinant. All right, number 9, true or false? Some mutations in just one single nucleotide can cause devastating effects in an organism. Is that true or false? Can just one nucleotide cause a devastating effect? So yeah, I'm glad you guys remember this one unanimously. So this is absolutely true. It can just take one nucleotide and then throw off the whole function of the protein and be devastating for the organism. So gene therapy is what? Okay, so gene therapy is a mechanism that we use, basically it's a term referring to the means to change somebody's like genetic mutations. So any genetic diseases are what we'll be treating with these. So that's what gene therapy is used. So we're actually doing therapy on their genes. Let's look at our little podiums real quick, and then we'll kick it into chapter 9. I just want to make sure that I give you credit for joining. Okay. That's easy enough. More. All right, good job, guys. I hope that was kind of helpful. All right. This one.
Question. For
Yes.
the group project?
Yes.
If we did like a PowerPoint or Kahoot last time, are we allowed to like do the same?
Yeah, you can do the same type of thing. I don't mind, especially if you felt like it worked well with your group. Absolutely, that's fine. If you want to try something different, you're welcome to. And if you want to try something that's not on the list that you think could work well, just let me know and we'll talk about it. But yeah. Okay, so before we get into chapter 9, okay, first, trying to figure out what I need to talk about first. Sorry, you can tell that it's been a lot. Okay, so let's go over kind of what is due because we've got midterm coming up. Yeah, like literally, like when I say midterm, I not only am referring to like the lab midterm, but also like the fact that it is the middle of term coming up very soon. Yes. Yeah, it's crazy to think about. I don't know how it happened. I feel like it's only like the second week of school still, but I feel that way every semester. So I'm going to talk about things that we have that are due in like lecture versus lab so that we're all aware of like what things you should have been paying attention to. I feel like if I list it big picture here, it's easier to see it. There's only so many ways I can put it on Canvas. It's just not as visually helpful. So anyways, lecture, we obviously have attendance. This is a participation. thing for you guys. My online class does not have this because obviously they don't attend lecture. So what they have for their participation credit, since we don't go over the Kahoots actively together either, they do those separately and then just show me evidence that they have done them. So that's how they get their credit. If some people who have not been able to make it to class, if I either have them to get your participation, because if you are missing class and you're willing to make it up, I'll work with you, right? Especially lecture. It's harder with lab because I can't necessarily give you that absence back. That's just a policy. But in lecture, if you're willing to watch the lecture and find one of the questions we go over in class, if we have them that day, then you can always like tell me what the question was, what the answer was, but you also have to tell me why that's the correct answer. So that's kind of taking a little bit further to just be sure you're paying attention. You can either do that, or sometimes if we don't have questions, I'll have people just do the Kahoot and show me the evidence. It's like we do with online. So those are always there as an option for everybody. If you happen to miss, of course, if you're missing a lot of lecture, I mean, that's on you, right? So you can still get the points, but you just have to contact me about how you want to get them. And if you're just missing a whole lot just because you don't want to come to class and you'd just rather do it at home, please talk to me about it first. Make sure that that's okay. Otherwise, I'm just going to assume that, I don't know, you're getting lazy or not getting up on time or whatever. And I don't want to, promote that. But if you just feel you learn better at home for some reason, let me know, we'll work something out. Okay. Anyways, that's your attendance points. Then we have, which is just participation based, right? This is for showing up. The fact that you guys came here and you're going to participate in the questions if I call on your name. Then we have the chapter quizzes. I'm going to put this chapter because I forgot to write that chapter. Which are due before each exam. We learned about those. We at least familiarized ourselves with those for the last for Unit 1 exams. So now we know those are going to be there, hopefully. We have our unit group projects. There's 3 of them. And then there's 1 individual project, which is for Unit 4, which will present on your individual disease, which you guys forgot. You can always find that one on under the group project, still under like unit projects. But yeah, it's individual. You have to sign up for your disease beforehand. And then it's just make a presentation about that disease. It has everything you need to put in there, on there. And an example of a particularly good one that I have kept for many semesters now as an example. So anyways, these are your so unit project and then the group grade, which again, those are due at the Unit 3 because that's the last group project that we're gonna have. So that'll be due then. Those won't really pop up on that. I try to update the group grades for those so that they will still signal you at each unit project. And I haven't updated it for this one. That's okay though, because you guys kind of get the gist of it, but we'll get to it. All right, we have 3 unit exams. Unit 1, Unit 2, Unit 3. They are not comprehensive, those ones, okay? So when we have Unit 2 coming up, even though it does fall in the midterm, it is only over Unit 2. Okay, and if you didn't see the announcement I made, it kind of says that in there. All right, and then we have 1 final exam, which is semi-comprehensive. What I mean by that, can't write today. Okay, semi-comprehensive final. It will be 100 questions. I think I've mentioned it before. It's still all multiple choice. 100 questions, it's a final at all, right? Multiple choice on the computers again. 50 of those questions will be focused on Unit 4. The other 50 are going to be Units 1 through 3. So semi-comprehensive, right? So there is a focus on Unit 4 with that one. All right, for the lab, hopefully I can fit all this. We have pre-labs, which are due before we go to lab. If you are late with them, you can still take them for delayed credit, but if you're taking them late, like as in after lab has started, every time, at some point, if I feel frustrated enough to get on to you, you might be hearing from me saying like, no, you're supposed to have these done. You don't come to lab anymore and you're going to get an absence until you can get on board with, because that's in the syllabus, right? It says that you don't come to lab and you'll get an absence if you don't do it. I just don't enforce it usually if it's just an accident now and then. We have our in-lab pictures that we have to upload. We have our lab quizzes. You guys haven't been paying attention. There are lab quizzes that are going to be due by the lab midterm. I scheduled it that way to try to give people freedom, but more or less it just comes out as like people didn't know they were there and they forgot about it. And now suddenly these things are coming up, like I was trying to hide it and I wasn't because, hey, surprise. It was not only on Canvas, but it was also in your syllabus. So not a surprise. It wasn't meant to be. I just was trying to give you guys freedom in how you wanted to get it done. If you want to do them after every lab, great. If you want to wait till right before the lab exam as like a test to see what you know, great. Do what you want to do with that. Just be sure it's done by then, okay? Then we have the unknown project, which we're kind of doing throughout, and we'll get into this more as we get more results. Some of you have already asked me about filling it out. That's cool. If you need help with that, just let me know. Then we have the midterm for the lab. This is a practical. It will be done in lab. It will be on Scantron. You will need number 2 pencils. I'll give you the Scantron and you'll go around to, you don't need to bring the Scantron is what I'm saying. Do bring your own pencils. I don't have so many to hand out, but we'll go around from station to station. There's usually about 2 questions per station, sometimes 3, sometimes 1, just depends on station. Overall 50 questions, and they're all multiple choice, all of them. So only A through E, because that's what the Scantron affords for. So there's not going to be like a million things to choose from. And right now, the way I have it set up, there's not going to be like, if there's a more than one answer, you choose more than one. It can only be one answer. So hopefully that's helpful for you guys. But yeah, you won't have to make slides. You won't have to use a microscope. You'll just have to look into it and tell me what's going on with it. Like recognize that, okay, I look at this and oh yeah, this is an endospore stain. And now you can say, this is an endospore stain. The first stain was malachite green or whatever. You don't obviously write it, but you select it on your sheet, right? So anyways, that's what to expect from that. And we can go over it in detail more, a little bit more as we creep closer and closer. And then we have in lab, one last thing, which is a comprehensive final. Again, I would call this semi-comprehensive, and we'll go over more on what that means when we get to the final. It's just a little early to be doing that. But you will see, absolutely, you will see microscope parts again, and you will see the stains again. But the other things that are kind of in the background, which we're about to get into, won't show up on the final. They'll only be on the midterm, okay? All right, the other last bit of stuff is extra credit. This will be when we have our in-class participation, your group question participation. The online class doesn't have this either, right? So what they get to do is actually kind of, even though you do this every single, hopefully we try to, every single class period, then they get the option of going back to one of the unit projects that their group didn't do. And they can pick another one and do that for that extra credit for that unit. And they have to get it for each unit. So that's what they get since they're not getting called on out, you know, by name in class and everything like that. So that's the trade-off. It's either get called on potentially every single class period, or, you know, they get to do an extra unit project by themselves, not as a group. Okay. So that's optional. And then we have completion. This is completion of everything in the course. It's just a possibility of 1% for each of these areas. You can only give 3%, where that's what we're capped at in the school STEM. So group question participation is 1% potentially up to, and we have completion of the course. This is a 0 or 100 thing. So if you have not done everything in the course, like if you have a missing assignment, you've got a 0 on something that you didn't turn in, you do not qualify for this. Okay. Get your work done. And if you're concerned about something that could still potentially be available, because some of this stuff can still help you for the final, email me and we'll talk about it, right? If you think that you could be, you know, able to get this. If you were late and you made up a thing and we talked about it, then you're still covered. So as long as you did that work, okay? And then the last thing is going to be like those little quizzes, like about the syllabus and whatever. Those give you a little bit of points, along with any of the evals. So the class evaluations that you guys do, the course evaluations that you do at the end of the semester, where you basically like write up about how you liked the course and what you liked about it or what you didn't like about it and everything like that. I don't get to see those until, I would say I don't get to see them usually until after the final, but I haven't seen mine for over a year now. I don't know what's been going on, but they haven't given them to us. So I have no idea what people from a couple semesters ago said about the course. I usually try to build off of that and I listen to it and I build off of it. But I don't know, since we've had the turnover with everybody, it hasn't really been catching up. But anyways, these are the things you can get extra credit for. 1%, 1%, 1% possible there, right? They will not, I will say, I want to reiterate about this because people get confused. They're like, well, I'm doing these things, I'm not getting points. Yeah, I bet you're not. These are worth 0% until your final grade posts. They are worth nothing. until your final grade post. Because what happens is, if I try to post these now as worth credit, there's no option on the Canvas gradebook for extra credits. So it counts it as a, like an assignment. So if I do it that way, it's gonna bring your grade down if you haven't done them. And that's not fair, right? So we aren't gonna post them, so just keep track of it, make sure that you are doing the thing, and you should be getting them. If you're concerned, let me know and I'll update it. So I just wanted to go over that with you really quick. The other thing before we jump into chapter 9, because I'm going to try to make this one and chapter 25 as quick as I possibly can, even though they're both chapters. This weekend I was supposed to go to Boise for my cousin's wedding, and I've been, like I said, really stressed out working on stuff for work. And I'm at home and I'm like just really tired. I'm like so tired that I'm having to sit down and stuff. And my husband's like, maybe you're sick. And I was like, no, I'm just tired. I have like stress. Like, you already know that, man. You keep getting freaking texts from Tim all the time. So, so he's like, cool. But then why are you hot? Like, why are you all so hot though? And I'm like, what do you mean? He's like, go take our temperature. I'm like, you're just trying to get out of this. I was like, I had an argument with him about it. Like, you're just trying to get out of this. You don't want to go. Like, I didn't really want to go either, but like, we'd already paid, and a lot of it is non-refundable, or you can get it back as like miles, but like, it's a lot of money, so he had to go take a temperature. So I took it and it was 101. Ooh. And so I was like, I guess we're not going, because you're not supposed to fly with a temperature. Like, they very strongly advised against it. Not to mention, like, what if I had gone and I started feeling worse, which is what happened this weekend. I started feeling a lot worse. I know. Insurance won't cover it while you're out. Right, exactly, right, exactly. Like we were talking about, exactly. So anyway, so I started feeling worse. I'm really not good today, but I want to be sure. I have a lot going on today. I need to be here. So it's not contagious. What I have is a kidney infection. Oh. And I'm on antibiotics now, but I just started them yesterday. So I'm on the antibiotics and I can't ever send the azo pills, the orange pills. You guys know, right? So I'm on that and we're trucking through, but I've still got, like, I can feel the fever and I just feel it coming out. So we're cranking through chapter 9 and chapter 25. You're welcome. We're going to hit the things you need to know for the exam. And we might go a little over some stuff as well, but we're definitely going to hit the stuff on the exam. And if we do not cover it at all, guess what that means?
Won't be on the
not on the exam, right? Yes.
About to ask this earlier, are the lab midterm and lecture midterm, are they both going to be on the same day?
No, they're not.
No, okay.
They're like a week apart or something like that.
Okay.
Or so, something like that. Yeah. I try to separate them and not be on the same day because people complain about that.
Are they like October 6th?
One of them is like October, like 4th or something, 6th or something. And then the other one is like mid-October. I don't remember what day exactly. 15 or something. The project,
like for this one.
do you guys remember when the project was due? The 2nd? Yeah, it's next week. Yeah, for sure. Yeah. Sorry, it's on the announcement. If you want to look at it, Grace, if you haven't had a chance to, I posted an announcement about those dates.
I haven't really looked into the project.
You're fine. You're fine. Okay, so let's so I'm at home and I am not in Boise, and I kept getting emails from Tim about let's start the revolution, and I emailed so-and-so and I emailed so-and-so, and then I've got the freaking president emailing me, and I've got like all this stuff, and I'm like on like the couch like just trying to hold it together. And then it was just a lot. So it's been a really busy weekend, even though I didn't need for 2. And then plus I felt so bad because I didn't tell Dr. Rhodes, because he's the department head, that I didn't go. And he'd been talking about how excited he is. He really likes Delta Airlines for some reason. He was like talking, he was talking about Delta Airlines and about how like it's a special screen in Detroit. If you guys don't know what I'm talking about, it's actually worth looking into. Like, they'll scan your face and everything at TSA when you're getting checked in or whatever. But that facial recognition goes on throughout the airport. So you can be looking at a screen and it'll have your name and like what gate you need to go to and point you what way you need to go. And it has a whole bunch of like basically facets to it. So if you're standing looking at it, it'll show your name. But if your brother, like his brother was standing next to him, he saw his name on the same screen at the same time. Yeah. So that's really cool. But he was telling me all about that and just having looking pictures of it and everything. He's like I was like on Sunday, and he like sent me this article from I don't know, the New York Times or something about how Boise is the next, coolest city in America or something. I'm like, great, because I'm sitting here in freaking Bethany. And I still haven't told him about it because I feel bad. But I was talking to somebody, I don't remember who it was, saying about how like, I was like, I'm not sure if I'm gonna go in today, like, I'm not contagious or anything, but I just don't feel great. I have all these things I need to do. And I was like, I'm sure though, that the class wouldn't really complain too much because they still have to have the lab, because I can get you out of the lecture, make you watch the online lecture, but can't get you really out of the lab, which means that always the only other person that can cover my labs, Dr. Britt. So I was like, I'm sure. He's popular with the ladies. If you guys haven't met him, he's very popular with the ladies. Anyway, so that's, I don't know, that's my weekend. So chapter 9, so let's get into that. Chapter 9 is going to be talking about nutrients and basically how microbes are getting their nutrients. The biggest issue with like what you need to understand about the nutrients, of course, is the stuff that we are building, all of these macromolecules. So CHOMPS is the acronym that the book uses, and I thought that's actually a pretty good little way to remember it. It's just carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So literal, actual chemical symbols for those things are the most common molecules you're going to find in cells, and what you need in your nutrition somehow, somewhere. The book goes into detail about where you can get each of those nutrients and everything, but some terms that you do need to be aware of for chapter 9 include your carbon sources versus your energy sources. Okay, if you are a heterotroph, you have to get your carbon in an organic form. What this means is that we are heterotrophs, right? We have to eat something that already had organic carbon molecules in it. We can't get inorganic carbon, like carbon dioxide or carbon found in rocks or something, to make our carbon-based molecules, right? We have to have like organic molecules already. So we're heterotrophs. So the other opposite of that is the autotroph, which is like plants. And some of our like cyanobacteria, just FYI, and our algae, if they're using inorganic carbon, like carbon dioxide, and some of them can even use things like, limestone rocks and whatever. And more important to me is that you understand about basically the photosynthetic versus the non-photosynthetic, like the main groups, right? So the photosynthetics, they get inorganic carbon dioxide and fix it into an organic form so that they can use it for their cells, right? That's where they get their organics from. They make them for themselves. Cool, right? So they're doing that using the sun and the carbon dioxide in the atmosphere, and then we can go for the things that we eat. We can go and eat the grass or whatever it is, and then get the organic molecules they built, right? So it's pretty cool how that can work. So they build their own organic molecules from scratch using carbon dioxide, right? You guys remember growth factors. This is anything that's required in your diet that you can't make for yourself. This goes across the board for any organism. If it can't make it and it has to have it in its diet, it's a growth factor. All right. Also, okay, so this gives us to the energy forms, which I think is like, we talk about it specifically. Yes, it is. Okay. The energy forms. So we talked about the autotrophs versus the heterotrophs. That's the carbon sources. So think about that. That will come. I'll summarize it here just a second. Just so I know we're on the same page. But now we're talking about where you get your energy, which is a different thing because the phototrophs get their energy from photo light. That's where they get their energy from. We get our energy by breaking down the things we eat and turning it into ATP, right? So they get their energy from the sun to build into that process. And then we get our energy from the things we eat, our chemical compounds. So that's why we're called chemotrophs, because we break down chemicals. All right, so those are the two terms. So there's some ways you can combine these that I feel like are kind of obvious. Autotrophs get their energy from the sun. They're auto, so they get their carbon from inorganic sources. So it has both right there. We can even break it down, look at it like this. Photo, energy, if you guys don't remember, the symbol means energy from sun. Auto means carbon from inorganic. I say inorganic, but really for our purposes, the only thing I'm ever going to ask you about is whenever that's carbon dioxide. Okay. There are other things that can fall in here. I just don't give a crap about them, right? So neither do you. All right, so that's the photo autotrophs. Remember, photo, energy, auto, carbon. It's a different thing. All right, then we have the chemo heterotrophs. Hey, that's us. So we get our energy from breaking chemical bonds. And then hetero is referring to where we're getting our carbon from. So we get our carbon from organic sources. So it has to already be organic, is what I'm saying, right? And where do we get organic from? If it has to already be organic, from the other guys, right? Because they're the only ones. Or other heterotrophs, right? Because you can be a predator and eat other organisms that already ate the other grass or whatever. So this is the idea. So yes, this is getting into that concept of the food pyramid, but breaking it down in a little bit more nutrient level, right? But this is what you need to know about this part of it, okay? We're going to get to aerobic respiration in chapter 10, right? So we're moving on. The aerobic microorganisms, in case you guys don't remember what these guys are, saprobes are any organisms that are involved in breaking down like dead, decaying matter. It's not just going to be fungi. Bacteria can play a part in this as well. So saprobe is a term for anything that breaks down dead things. That's what you need to know about them. I don't need you to know about how it happens either, but they do release enzymes outside of the body to break down the bigger things into small particles that they can then absorb. Okay, we're not going to talk about obligate versus facultative on this part. Cool, they can need to be that way, or they can just switch between. Nice. Nutrient absorption. Okay, this is going to get into diffusion and osmosis. The next part. So we've got down our energy versus our carbon. We know what a saprobe is. So now we're going to talk about osmosis. And that we have to talk about diffusion first to get into that. And then, of course, we're going to talk about tonicity. That's our next big kind of concept, not the biggest from this chapter, right? So diffusion is movement with a gradient. So that is to say, if I have a big old pen over here that's all netted off with a bunch of bouncy balls in it, right? And I cut that net open or release it open the door, you know, the floodgates go, they're just going to spill everywhere, right? That's diffusion because we had a bunch over here, none over there. So we're going to move so that it's equal on both sides. So that's all diffusion is. It's going to happen naturally. It's driven basically by physics, essentially. It's not like a philosophical concept so much as it is like physics. So luckily, we don't need to know any math about that, and that's great. So, because I hate physics, but that's all diffusion is. It's trying to equal it out. Okay? If there is a whole bunch of bouncy balls over here in this pile, and there's just a couple over there, are those bouncy balls going to naturally just come over here? Absolutely not. Right? That's against the gradient. So we don't do that, right? So osmosis. Osmosis is diffusion of water. Very specifically, diffusion of water. And we usually are talking about it in the terms of movement across a selectively permeable membrane, because this selectively permeable membrane is essentially what all cell membranes are. Okay? The cell membranes can allow water and other nonpolar molecules to cross, but other things can't cross just on their own. So that means that if you're sitting in a solution of salt and you need to equal out what's going on outside and inside, or if the salt is forcing you to give up some water, right? Sucking out that water out of your cells, only water can move. The salt can't move in naturally anyways, okay? Not across just the membrane. So we're going to get into that, but osmosis is diffusion of water across a membrane. I'm sure you guys have seen stuff like this. If you're putting your little baggie into a water that has this much of crap in it and that much of crap in it, and is it going to have water move in or out? Only, remember, only the water can move, nothing else, right? So you're trying to balance out concentration outside of the cell versus what concentration is on the inside of the cell, which is important to keep in mind, okay? So in isotonic conditions, this is when we're equal. They say the concentration of the solute, which remember, we're back to the beginning, solute is the crap dissolved in it. So let's say salt, okay? Concentration of salt outside is the same to the concentration of the salt inside. We're not talking about like you put a tablespoon in here and we need a tablespoon in the cell. We're talking about the concentration overall in that solution right? So if you have a giant C that is the same concentration of salt as what's concentration inside of your cells, then you're good, no matter how big that C is, or if it's a tiny cup. So when this happens, when we're equal on both sides, we are not just chilling. The water isn't just not moving because of course chemistry, right? Because I don't know, physics and stuff like that. But the water molecules are still moving across. They're just, we would say, at an equal rate, in and out, same rate, same amount. Okay. All right. Hypotonic, and this is where we get big, right? So remember that ISO is going to be equal. Then we have hyper and hypo. Okay. So starting with hypotonic, right? So tonic is, think about it as a tonic. The liquid the cell is sitting in. So the liquid the cell is sitting in is hypo compared to the cell. Okay, that means lower, right? So if we look at it that way, that means that our cell, let's say it had our cells, they're not 100% water inside. Obviously, we have things like proteins and salts and all those nucleotides, all sorts of stuff. So if our cells, let's say, are 95% water, and I put our cells into Dasani, and it's 100% water, well, it has some minerals, but let's pretend, okay? 100% water, okay? So which one, the cell or the water that it's sitting in, has the most solutes or crap in it? The cell does, right? So the cell does. So it's going to want to, because we can only move water across, right? We can't move the crap in the cell across to equal it out. So what's going to happen is, even though we're the crap in the cell with all our little bouncy balls is our crap, okay? And outside is no bouncy balls at all. What we'll try to do is take in more of that water to dilute out our bouncy balls until they're the same concentration as outside, which is easy enough to say if there's just a little difference. But when there's more than a little difference, like 100%, and you're never going to dilute out to 100%, right? Then you will swell up with water, trying to dilute out your insides, more and more taking the water, dilute, taking the water, dilute out my bouncy balls until there's none is the goal, right? But I don't know because this is physics. No bouncy ball concentration over here and the no bouncy ball concentration out of there, because we want to be ISO. So we'll keep doing that until we get that, but we can't. We can't. It's impossible. So we will burst the cell doing that if we don't have some sort of protection with our cell. The cell wall can provide that protection. So that's a possibility as well. So that's useful for some cells to prevent that bursting. So that bursting is also called, remember the term for it, right? Lysis. Lysis. All right, hypertonic. So now we know that this is hyper, so that's more, right? So the tonic, the outside of our cell, the stuff that we're sitting in, is hyper, more than what's in the cell. So now we have our little cell with just a couple of bouncy balls playing over here. And outside of it, it's just a ton of bouncy balls, just stacked up, packed around us. That's like, let's say, salt, right? And we all know what happens if we pack ourselves in salt. It's going to draw the liquid out, right? Why is that, though? That's because nature, diffusion, osmosis, is trying to, like, basically bring equilibrium there, the ISO. So remember how we were bringing it in with the hypo to try to water us down? It was impossible. Now we're trying to water that down so that we're equal. But we're just a tiny cell. So are we going to ever be able to do that either? No, right? So there's extremes there, of course, that I'm giving you examples of so we can see the big picture here. But of course, you have just like little differences, and the cells can manage those. So with the hypertonic, we're trying to water out that hyper to make it ISO. And we will give up a lot of our water from inside of our cell in doing that, and then shrink everything else down inside of our cell, a withered old cell. Bacteria can manage that as well with their cell wall. It gives them like a structure to stick to maintain that shape, especially the gram-positives. They're a lot more stable with that situation. However, at some point, most bacteria are going to have to give in to that and die, basically. And that whole situation is called plasmolysis or plasmolysis, right? The one before was just obvious lysis. We're just going to burst it open. And here, we call it plasmolysis because all the liquid in like our cytoplasm is gone. So we lost that. That kind of left us. That, in a way, was what lysed, and our cells left over. Little wrinkly little guy. So we can use this whole system to preserve food. That's why we pack things in salt or in sugar. Has the same effect. So this is called osmotic pressure to force water out of the cell to limit the growth of microbes. So that's why we pack things in salt. It's why whenever you make preserves, we have high sugar content. It has the same effect. It doesn't have to always be salt. Anything that has that concentration has that effect on the cell. So it can be used for osmotic pressure purposes. This is not going to sterilize anything by any means, but it does help, reduce spoilage. All right, so here's some pictures of what I was talking about. This is a great thing to come back to and refer to. Me just describing my scenario with bouncy balls and stuff is great, but now this is going to put it in context of the cells. Highly recommend coming back to this figure in your book or in your text or in the slides, whatever works for you, to refer back to which one is which, okay? All righty, in the environment, cool. Cells have developed like mechanisms basically to survive in the conditions they have to survive in. So if you are a cell that lives in fresh water and is going to be hypotonic around you, right? They have means to get rid of that. So they can either, the water that's rushing in that normally would bust them open, collect them in vacuoles and transport it out actively the whole time. Or we can, that's what amoebas will basically do the whole time. The bacteria just have the cell wall to keep the shape no matter what. So let's just hold it in the whole time. But the amoebas can actively move it out, but it takes a ton of energy to do that, right? Because that's against the gradient. Right? Anytime you're moving anything against any gradient, that's going to be requiring energy. It's like walking uphill. I mean, can you go down a hill without using any energy? Yeah, you can sit on a little scooter and just let it carry you down. It doesn't cost you any energy to go down the hill. So, but going up the hill against the gradient, you can't do that, right? You have to expend energy to do it. So it's the same way with this stuff. So if we have hypertonic environments, like a whole bunch of salt on the outside, if you're something that lives in one of these extremely salty lakes or something like that, they have the ability to take in salt on their inside. So if they have a lot of salt on the inside and a lot of salt on the outside, they can live like that. And that's fine for them. That's what works for them. But if you take those cells out and put them in the normal environment of what other cells are experiencing, that would kill them because they're not equipped for that. They have salt in them, bad for them. So they're adapted for that environment. We call them halobacteria. Archaea are also pretty good at that. So we have simple diffusion, which we already kind of know about. That's going to be movement with the gradient. The gradient is what drives you. We have facilitated diffusion. So we can't just move anything across the cell. Some things we need to get into the cell. So we give them a little channel to go through because they can't cross that membrane by itself. So we give them a channel, but it still goes with the gradient. That's facilitated diffusion. No energy is being put into that. We're allowing diffusion of those molecules, just they can't cross the membrane otherwise. All right, then we have, well, I'm not going to get into this too much, but obviously you only have so many channels on a cell, so that can sometimes limit your transport abilities. But active transport, obviously energy is going to be required for going against a gradient. Maybe you just need a whole bunch of something in the cell, more than what there's concentration of outside. Or you're trying to get a toxin out of the cell. Obviously, you're trying to get to zero toxin in the cell and all of it on the outside. So if that were the case, then now you might be fighting against a gradient, in which case you have to have energy. And there's a word for what we would be doing to do that, fighting against a gradient. That's called active transport. Active, we're active. Do you need energy to be active? Obviously, yes. Yes, and this keeps the concentrations. It says the potassium, sodium, and the hydrogen ion. Some of these things like potassium and sodium are very important in sending nerve signals. So you always have to have that potential saved up in order to send signals with your nerves. And sometimes you have to go against the gradient to repack that or to make it happen. So we need to use active transport for that. When we talk about our hydrogen ions, we're going to talk about aerobic respiration in chapter 10, and that'll make way more sense with that later. But all right, so we need energy in order to drive that. When we talk about endocytosis or phagocytosis, phagocytosis is a type of endocytosis, just big time. That's using a lot of energy to move the membrane around something and create a vesicle around it. So that's big time, moving something into the cell. So we have that, and we have cell drinking, which is pinocytosis. I always think of Pinot Noir or something like that. I don't know, Pinot Grigio, whatever your favorite is. That's what I think of. So drinking, so that's cell drinking, like oils and things like that that normally won't move into the cell. You can take little sips of it and bring it into the cell as needed. But phagocytosis is our biggie. There's a lot of things that are important for like microbes to live. Things like temperature are important. Cool. Watch me do this. It's not on unit 2. Okay. You guys already know about pH and the importance of that and like anything. Things that are able to deal with some of these other things. If you're interested in this stuff, of course, go back and look at it, but we're not going to have this on to unit 2. So now here we are at a thing that we need to know. Symbiosis. Okay, so this is a pretty good little chart that breaks down our possible relationships between organisms. The ones that you will be tested on for the exam will be the symbiotic ones. We all know that things can affect the lives of other things, but we aren't necessarily relying on that all the time. Things like you have to eat, I don't know, food. So any of those things that you eat, great, and you have to eat it, but like it won't always be this thing or that, right? However, there are some situations in biology that we are aware of that you have to have that relationship. One organism does or the other one does. Okay? And that's what symbiosis is about. Organisms living in close nutritional relationships that is required by one or both members. Required. All right. There's 3 types: mutualism, commensalism, and parasitism. You do need to know these. You'll be asked to know which one is which, essentially. Mutualism exists whenever organisms live in obligatory, they have to live with one another, but they're both benefiting. They're both getting some sort of benefit from one another. So this might include a lot of the microbes that live in or on humans, like in our gut. So the gut bacteria in your gut right now, they can break down foods that your body otherwise wouldn't be able to and make it so you can use those nutrients. However, at the same time, they're getting protection and nutrients for themselves, right? So we're both benefiting. That's mutualism. Makes sense. Mutual. We're mutually, benefiting from this. All right, commensalism. This is one member is benefiting and the other isn't, but they're not harmed. Just one is benefiting and the other's just chilling. All right. I don't really have a good example, but the term for the one that's receiving the benefit is the commensal, and the co-inhabitant is the neither harmed nor benefited. And I don't know what else to say about it. Trying to think of a good example, but I don't really have one off the top of my head right now. Anyways, parasitism, this is an obvious one. And I feel like these kind of go like obvious, like you're either both benefiting, only one is benefiting and not harmed, or one is benefiting and is harmed. So those are our choices. So parasitism, the parasite is benefiting, and while it is benefiting, it is harming the host. We kind of know that about parasites. This is obvious things like we would already consider to fall into this category, like ticks and fleas and stuff like that. You can always think of those. And if you think is something living with this organism like that, if the answer is yes, then it's also probably a parasite, feeding off of that host and causing harm to it. You can see how a lot of pathogens could fall into this category. So things like the plague itself, and I'm not talking about the fleas that bit the person to give them the plague, but the bacteria itself is benefiting by living in you, but also causing you harm, right? You're not getting any benefit from having the plague. So you can look at it that way. COVID is getting benefit from living in your cells, and you're being harmed. Parasite. Okay. Let me just click through all this stuff to make sure I'm not biofilms. We talked about that. Binary fission. These are kind of important. These aren't on the exam, but I do feel like they're going to be on a later exam. So I do want to touch on them just a little bit for now. When bacteria grow, they have 4 phases in the growth curve. They have the lag phase, the exponential or log, which is logarithmic phase, and then the stationary phase and the death phase, okay? Pretty straightforward. Lag phase, you just introduced your crap to the culture. It just got in there. It's like acclimating, seeing what kind of nutrients are available, and it hasn't really started actively dividing yet because we have to start getting our resources together to start even copying our DNA and everything like that, right? Lag. In the exponential growth phase, now we're actually actively dividing a lot, exponentially / logarithmically. In the stationary phase, now we're kind of running out of resources. So we're not dividing as much because we just don't have the resources available to. And we have a little bit of die-off. So maybe growth and die-off is starting to equal out, as well as just kind of stagnancy. Okay. Whereas once the death starts overcoming due to waste, due to lack of nutrients or whatever it is in that population, then we have death kind of winning out and eventually everybody dies. Pretty straightforward, but just remember those kind of 4 phases, okay? It goes through each of these in the following slides, explain them in a little bit more detail. Okay, so no questions. Let's do chapter 25. Sorry. Okay, chapter 25 dealt with microbial ecology, which is like the most boring thing to me in the whole world, but it has to do with like them being able to survive in certain kinds of environments and terms that we would apply to them. Turns out I am not going to really go over any of these biogeochemical cycles, but they're maybe later on in the final, I don't know. But we should more be interested in carbon and nitrogen when we look at these, okay? The carbon cycle is very simple. We have plants, we have CO2. The plants use CO2 and turn it into organic stuff, right? That's where it gets their organic stuff. And then we or other organisms will eat them. So we can eat them. This is we or other organisms, by the way. So this is just microbes that we're talking about. Or we can die, the plants can die, and we can break it down that way. Doesn't matter, either way, you're gonna involve it in creating energy, ATP, via aerobic respiration typically, and give off, what do you think? See how easy that is? Like, I feel like it's not even that hard. And then there's other things like volcanoes or industry that have combustion, natural or unnatural, that can also contribute to that carbon dioxide in the environment. There you go, carbon cycle. Okay. Obviously, when you ingest organic carbon, it can become part of your body, right? So that's what I want to be clear about. But if you are somebody who wants to argue with me about global warming and whether it is real or not, don't try. How much it has to do with carbon dioxide or methane, that could potentially be an argument, potentially. But global warming itself, regardless of that, is a real thing. It absolutely is happening. And you can look at it in any chart whatsoever. But this is looking at our carbon dioxide emissions since 1960, basically like since we started having booming production of carbon dioxide from industry, essentially, and cars and all of that. So this started happening way earlier than that, back in like 1900, in the Industrial Revolution, but got big after that. So you can see quite an increase since then. Cool, you already know that probably. Worse than carbon dioxide for our atmosphere is methane. And methane is usually made by farts, just being honest. But the methane in the farts, let's be clear here, come from the bacteria in the gut making the methane, right? So the cows themselves aren't making it, or we aren't making it, but the bacteria or archaea in the gut are making it. And these organisms can also live outside of guts. They can also live in things like swamps and stuff like that and release methane that way. And it seems that the environment is favoring those types of organisms. Yes.
There are some theories that there are microbes on Mars because there is a certain amount of methane in the environment.
Yeah.
But that could also be just like natural, like volcanic activity.
Yeah.
But we don't know yet. I
Yeah,
just thought
it's
that was
hard, yeah.
interesting too. There might actually be Martians, but they're just
It's so hard. I feel like every other year it's something else, right? Like they're like, oh, there's life on Mars because of this. And I'm like, yeah, cool. When you can prove it though, like come back to me then. Like
I hope there's life on Mars. That'd be pretty cool.
it would be. Also, I don't know how I feel about it, right? Because I just feel like you don't know what those organisms are going to do if they're exposed. How do you prevent microorganisms that you've never seen or encountered before from coming back to Earth? If you come back.
True.
And how do you know that they're going to be? Anyways, I'm getting into horror genre now. So, so I'm a big horror movie fan. So anyways, nitrogen cycle. Really, there's all these terms that we can apply to it on how you can deal with nitrogen in the environment. Nitrogen fixation, ammonification, nitrification, denitrification. I will never have you memorize what happens in each. Just know that these are terms, the 4 terms referring to the nitrogen stuff. Okay, these are 4 ways that we have in nature that have a cycling of nitrogen in organisms. That's the terms for them. That's all you need to know for them, okay? Now we could need to know, we could go through these cycles, and in fact, these slides do go through these cycles, but I'm not going to even, I don't even like it. I don't like it myself, so we're not really gonna do it. But the importance of these guys, and I've mentioned it before, the plants cannot use the nitrogen available without the bacteria to change it into the form they can use. So if the bacteria in the soil aren't kept alive, then the plants can't grow. And that's a big issue that we've been having with global warming, just with the warming, not with the carbon dioxide levels affecting that, but just the hotness of it killing off, bacteria that are in the soil because they can't survive in that heat. There's not made for it. There's a reason why deserts don't have things growing in them. But yeah, we could eventually see a lot more desertification, as they say, on Earth. The rhizosphere is just talking about all the organisms and how they work together. We also have water on Earth, and if you didn't know that, there's water. There's water in all over the place. There's fresh water, and there's the oceans. The plastosphere refers to the amount of plastic in the ocean. 88% of the ocean surface is coated with microplastic particles. 88% of the ocean surface is coated with microplastic particles right now.
So yummy.
So we can have,
Sprinkle some in your instant ramen.
right, well, I mean, yeah. So we have microbes that, basically, okay, the plastics can create, like it says here, reefs. It's like the plastics can congregate together, and then we can grow on these reefs or under these reefs. It creates basically an area for microbes to grow where they shouldn't be concentrating otherwise. This can block out sun beneath, and I'm talking about, these will create islands the size of Texas. So some of them, there's at least one that size. So this can block the sun. It can create weird biomes that shouldn't be there, and it affects the ecosystem of the ocean overall, obviously. So like, I don't know, bad. It's bad, y'all. Freshwater communities are also important. The plankton, like the phytoplankton, make a lot of our oxygen in the environment. But these guys can also be like, their population shifted by different amounts of nutrients in their ecosystem, which is basically what, if you've ever heard these terms, oligotrophic or eutrophic, and referring to a lake or a pond or something, it has to do with how many nutrients are available, of overgrowth of like algae, blocking out the sun of the things below. There's no oxygen left over in the water, and the things below will die. You can have turning of a lake as a result of that because of things dying, basically. And that can be remediated, but it takes a long time. Now we're getting to water stuff, drinking water and wastewater treatment, okay? In drinking water, so water that we consider safe to drink is called potable. And potable water, P-O-T-A-B-L-E, some people think it's potable, but that's not, it's potable. It usually has to be clear, like it has to look appetizing on top of the fact that it shouldn't like have too off of a taste, but ultimately the biggest issue with potable water, you want to be free of pathogens, 100% free of pathogens. So that's potable water. So that's the goal with our drinking water, okay? So here's how we basically do it. We take water from wherever aquifers typically, and then we put it through coagulation where we make any particles that are in that water larger so we can filter them out, sediment them, and then filter out what's left. And we can even do this a couple of times. Then we will disinfect the water that is left over, usually with UV light or chlorine can be added, stuff like that. We can bubble chlorine gas through it just to disinfect it and not stay there. And anyways, then it'll go sit and wait for you to drink it. It's pretty straightforward stuff. When we talk about water contamination, in many parts of the world, drinking water is also used as dump or solid or liquid waste, either actual sewage or just trash. And none of that's good, right? So people are bathing and drinking in the water that they're having sewage in. And this is, yeah, I mean, it's not that surprising when you think about this, especially like when we're thinking about areas like in India where people consider the water to be sacred, like the river to be sacred. And they'll go do sacred things or bathe in this water that is sacred, but then like upriver or downriver, people are dealing with their waste and stuff like, and this is the truth, right? And they also, it has been tradition to send bodies down the river as well. So this thing doesn't occur because this is part of, cultures, but as we've had improvements in water safety, this has become, people become more aware of it, I will say. All right, so what we need to do for, if you're gonna treat wastewater, like, for example, you flush your toilet and you need to treat that water before you release it back into the environment. Because when you flush your water into the toilet, it doesn't just go away and that's the end. That's not the end of the story, right? So we know they're going to treat that water, but where does that water go? Does it get treated like and get sent into your tap? Does it get treated and sent into just a reservoir that we never touch again? Does it get treated and sent into the river or the ocean or whatever it is? It's the latter, by the way, that one. So what we do first, we will separate out in a primary stage large matter. This is going to be the obvious things like plastic bottles, the bicycles and whatever else that got left in just wastewater overall, right? We'll skim off the big things and also the bigger chunks of things. So smaller suspended particles will settle, and then we will allow biodegradation by bacteria and all these things, and that's going to create sludge. So this is just stuff that isn't getting degraded by that first stage, right? And that can settle out, and we can take the water off the top of that. We can use the sludge to decompose it further and get more water out, or we can use it as like fertilizer and stuff later on, depending on what stage we're at with this, because this can go through that stage a couple of times as needed. The tertiary stage, we're going to filter and chlorinate, just like we did with the drinking water. Okay, so it's at this point now where we have removed any of the solids from it, and we can actually filter it, and then chlorinate it. And then we can use this to release into large bodies of water or use for watering golf courses and stuff, right? So not usually for consumption. And then the solid waste can be used for fertilizer and stuff. We wanted to look into wastewater surveillance because of a lot of things at first, right? Just trying to make sure the water was safe. But it became an issue again with COVID because it's a tiny little virus and it was making it through our wastewater treatment systems. And because of that, we were having, contamination of the drinking supply and all that. So this was an issue back whenever polio existed in the United States. And it does not exist in the United States anymore. But it is a very tiny virus. And if we could filter that out, certainly we can filter out COVID. And so we started employing that same mechanism to filter out COVID so that now your drinking water isn't contaminated with COVID from other people having it. So it's pretty interesting. That came back to be useful for us. If you did not know, polio was transmitted by water contamination. And so fecal contamination of water wasn't being treated properly. They just didn't know that because it's this tiny virus they hadn't really seen before. So people were getting polio by just drinking the tap water. So crazy. All right, we already talked about potable water. It needs to be not gross and it needs to be actually free of pathogens. There's a lot of pathogens that we might be concerned about, like Giardia, which causes horrible long-term gastric issues, Cryptosporidium, which causes diarrhea, and it is a eukaryote protozoa. We have Campylobacter, Salmonella, Shigella, Vibrio, which is cholera, and other organisms related to cholera. Mycobacterium species, not just tuberculosis, but including tuberculosis. Hepatitis A is a oral fecal virus, and then Norwalk virus, which is another diarrhea causing virus. So these are all concerns that we would need to be sure that are not in our water, or sure these at the very least. So what we do is we monitor indicator bacteria. And so you would want to know, okay, well, yeah, sure, look for the pathogens themselves, but wouldn't you rather know like that there's a propensity for the pathogens to get through before they get through? So that's the idea with this. So we use certain indicator bacteria, which are species that we can find in birds or mammals, that if they make it through our filtering process and they're otherwise harmless, then that lets us know that we've got a bigger problem, that other pathogens could make it through potentially as well. So the ones that we use are called coliform bacteria. You may have heard them called fecal coliforms because that's what they are. These are bacteria that are found in human feces typically, that we use as indicators of contamination. And if they get through, maybe cholera can get through, maybe these other guys can get through. Okay. So it's just normal E. coli, the non-terrible E. coli. Normal E. coli, Citrobacter species, Klebsiella. I'm not going to test you over those, but these are just things that will know what coliform bacteria are. They are usually gram-negative, but they're typically gut bacteria that are used as identifier indicator bacteria of the water purification process. let's see. Okay, the best way to look at these guys, they can take water sample from tap water or whatever and filter it literally through a filter, like a very fine microfilter. And then put that filter paper that was on there onto a petri dish and let it grow. And then count how many colonies are there. You would hope there'd be none, but there's a limit that is acceptable for every water source. So clearly the lowest, we can have the least amount present in drinking water. And then going from there, depending on what you're using that water for, can have more and more and more. If you're releasing sewage water into the environment, that can have the highest. So they'll look at that. But yes, there is usually not going to be a zero. Hate to break it to you. Yeah, we want to try to aim for that, right? That's the idea. So that's how this would work, and this is how this is the filter growing little colonies here on this guy. I want to say we did this in my lab. I worked in last, so yeah, you can apply this concept to like normal taps. Make sure that your tap specifically is safe for humans to drink your water. Like we would get water from like Applebee's, and Applebee's like sort of tap water they would be serving to people. And so they would have us test it and make sure it was clean and stuff like that. So it's not necessarily that it's always just going to be like the city, which should be testing for it, but there's a lot of pipe that it has to go through to get to your cup, right? Is the pipe running through your house or your apartment complex? Is it clean? Right, and so you would hope that that's not too bad, right? But anyways, we would test for that for certain like restaurants and whatever to make sure that they were not bad. Anyways, I think that's it. I think that's all I'm going to go over. We already know you can use microbes for food and fermentation. One Health, which I'm not going to test you over, just in case you're interested though. One Health is saying that if we can keep not just focus on people health, but the health of the whole world, like then keep this area of life and this ecosystem of life and all these things together, then the whole world should be healthy and synergistic. And that's the idea of what one happens. But that's it. So that's that. Let's go to lab where it's going to be hot. I'm already hot.